Bismillahirrahmanirrahim, and welcome back to yet another course. And this particular course is on the microbial diseases, and where I'll be focusing on different microbial diseases, the diagnosis, the treatment, and prevention of these microbial diseases. Now, we will start the course with an introduction to the digestive system because this particular course will focus on a variety of the foodborne diseases. So you should have an idea how the digestive system normally work and how these pathogenic microorganisms they disturb the normal digestive system. So if you define this digestive system, we will start with the definition. This digestive system is actually a complex process which turn food into nutrients, and these nutrients are then used by the body for energy, growth, and the cell repair. Now, the process of digestion also involves the removal of the waste that is produced during the process of the metabolism. Now, if you talk about the human digestive system that we will see, we will start with the digestive tract, and we will have detailed discussion on the mouth, the pharynx, the esophagus, the small air, the large intestine, and then the anus. Then we will focus on series of structures and organs through which the food and liquid pass during their processing into forms absorbable into the bloodstream, and we'll also see about the structures through which the waste pass in the process of the elimination. We will then focus on the other organs like the pancreas and the gallbladder, and these organs actually contribute juices which are necessary for the digestive process. Then we will have detailed discussion on the salivary glands. So this would be the next topic in the course. We will start the salivary gland with an introduction to the endocrine and the exocrine glands. Then we'll focus on the salivary glands and their types. And if you talk about the types of these salivary glands, that include the minor salivary glands, the buccal, the lingual, the palatine, the labial. They are the examples of the minor salivary glands that we would see in the course. Then we'll talk about the major salivary glands that include the parotid, the sublingual, and the submandibular salivary glands. We will focus on the acinar cells. These are actually the building blocks of these major or the minor salivary glands, and the acinar cells usually come in two flavors, the serous cells and the mucus cells. So we'll talk about them in detail, what are they and what are their functions. Then we will move on towards the composition of the saliva, and we'll focus on different components of the saliva like the water, the enzymes, the electrolytes, the antibodies present in the saliva. We'll talk about their roles as far as the function of the saliva is concerned. And then we will focus on an important disease of the salivary glands, which is known as the sialadenitis. And the sialadenitis is actually the inflammation of the salivary glands. So we'll see about the causes, the symptoms, the types, and the medication about the sialadenitis. Then I would focus on the foodborne diseases. So if you define these foodborne diseases, they are actually the illnesses that are caused by the ingestion of the spoiled or the poisonous food contaminated by the microorganisms or toxicants. And these microorganisms or the toxicants, they may occur or they may enter to the food during the food processing from the production to the consumption. So we'll start by defining the foodborne diseases. And then I'll give you a list of the most common foodborne pathogens produced by the CDC, the Center for Disease Control and Prevention. And the first example that we would see in the foodborne diseases, that would be the Campylobacteriosis. And this Campylobacteriosis is actually an infection caused by the members of the Campylobacter genus, most commonly by Campylobacter jejuni. So our focus would be on the Campylobacter jejuni, and the discussion on the Campylobacteriosis that would include the biochemical properties and laboratory identification of this Clostridium jejuni. We'll talk about the different virulence factor of the C. jejuni. These are just some of the important examples that we would see during the course of discussion. We talk about the quantum sensing and biofilm formation an important, you can say, virulence factor of the Clostridium jejuni. We talk about the flagellum of the C. jejuni, again a very important virulence factor. Then we'll focus on the sources or the route of transmission of these Clostridium jejuni, talk about the symptoms, the important diagnostic test for the identification of the C. jejuni. I'll talk about the treatment, and then in the end to cover the to complete the discussion on the Campylobacteriosis, we'll talk about a complication which is known as the Guillain-Barré syndrome, an important complication. The second example that we would focus on that would be the Clostridium botulinum, another important foodborne disease. We will start with the general introduction to the Clostridium botulinum. We talk about the biochemical properties and the laboratory identification of Clostridium botulinum. Then we'll focus on the pathogenesis of the C. botulinum infection. How these toxin, Clostridium botulinum, is going to produce an important toxin which is known as the botulinum toxin or the botulin toxin. We talk about that, how the toxin is produced in the food. We talk about the infant botulism, you can say a subtype of the infection caused by the Clostridium botulinum. I will talk about the laboratory isolation and the diagnosis of the Clostridium botulinum, the symptoms, the treatments, and the good thing is that Clostridium botulinum or the infection caused by the Clostridium botulinum, that is preventable. We can prevent that. So we see how we can prevent this infection caused by the Clostridium botulinum. The third important example that we'll discuss, that would be the Clostridium perfringens. Again, we'll start with a general introduction to the C. perfringens, the biochemical properties and the laboratory identification. We talk about the Clostridium perfringens disease, which is known as the Gase gangrene. Sometimes it is also known as the clostridial myonecrosis or simply myonecrosis. We will have a detailed discussion on the Gase gangrene. Then we talk about the important virulence factor of the C. perfringens. That would include the alpha toxin, the perfringolysin O, and the Clostridium perfringens enterotoxin, the important virulence factor of the Clostridium perfringens. Then we'll see about the diagnosis of the C. perfringens, the symptoms, and then the treatment and prevention of the Clostridium perfringens. At the end of the course, we talk about the typhoid or the enteric fever. We'll start with the general introduction to the Salmonella genus. The reason is the causing agent or the pathogen that belong to the Salmonella genus. We talk about the cycle of contamination of the Salmonella genus. We will talk about the subspecies Enterica, which include the typhoidal Salmonella and the non-typhoidal Salmonella. Then we will focus on the typhoid or the enteric fever, which is actually caused by the Salmonella typhi or the Salmonella paratyphi A, B, and C. Then we'll focus on the biochemical identification of the Salmonella typhi, how you can do that. We'll talk about the pathophysiology, a very important component to understand the infections caused by the Salmonella genus, especially the Salmonella typhi and the Salmonella paratyphi. We talk about the signs and symptoms, and the talk about the diagnosis, how you are going to diagnose the typhoid or the enteric fever, the different type of the diagnostic test used, and in the end we talk about the treatment of the enteric or the typhoid fever. So this is the whole you can say syllabus or the whole course content that we are going to cover in this particular course.